Good morning. Good morning. It's very nice of you all to be here today. My name is Donald Trump. I'm the president and CEO of Roswell Park, and it's a distinct pleasure to be here to be able to announce to you another great accomplishment of a wonderful team of scientists and clinicians here at Roswell Park. 124 years ago, Dr. Roswell Park, who founded this institute, noted that it took the combination of laboratory and clinical study and education to really attack the cancer problem. Our discussion and presentation today is a demonstration of the productivity and merits of such a combined approach. Dr. Park really defined the outline for the organization of the modern comprehensive cancer center such as Roswell Park. And what we'll talk about today is an example of a remarkable accomplishment at a clinical facility, a research institute, and a center for education. We're going to tell you today about work on development of a vaccine against cancer. This has been led by an outstanding team of investigators, clinicians, laboratory, and translational scientists. And it's also importantly been done in collaboration with a small manufacturing company, Biospherics in Alcoma, New York. This work has arisen from the Roswell Park Center for Immunotherapy, and it represents discovery, development, clinical care, and public-private partnership to accelerate progress. Without further ado, I'd like to introduce Dr. Kunle Odunsi, who is our chairman of gynecologic oncology and the director for the Center of Immunotherapy, who will discuss with you the clinical trial and the program that underlies this development. Dr. Odunsi.
Thank you very much, Dr. Trump, and thank you to everyone for joining us for today's announcement. This gathering truly represents a landmark event because we are launching a new clinical trial that will harness the power of the immune system to fight cancer. But before I go any further, I would like to acknowledge the contributions of some of my colleagues without whom we will not be here today. These are a group of clinicians and scientists who have worked collaboratively, creatively, and effortlessly to make this possible. And I would like to acknowledge the following individuals: Dr. David Hone, Dr. Chris Choi, Dr. Tom Schwab, Dr. Paul Wallace, Joanne Becker, Dr. Pratul Shrikant, and Dr. Junko Matuzaki. Thank you very much for your work. I would also like to acknowledge the vision and foresight of Roswell Park Cancer Institute's leadership under the direction of Dr. Trump and our Deputy Director, Dr. Candice Johnson, in investing necessary resources into this program to allow us to bring the latest and best possible care to cancer patients in our region. Without the team effort and without the institutional investment in resources, we will not be here today. So let me move on and address the details of this investigational clinical trial and also raise the question, what makes this unique? Why are we here today? So there are at least 3 unique aspects of this clinical trial. Dendritic cells, these are the cells we're going to use in this clinical trial. They are the gatekeepers of the immune system. And we have found a way to harness that critical role to fight cancer. First, we will take the dendritic cells, as shown here, and multiply them outside the patient's body. We'll take a patient's own dendritic cells, multiply them, and generate millions of cells. We will now take those dendritic cells and attach them to a protein called NY-ESO1, shown in red on this slide. we have a special way of attaching the protein so it binds very tightly to the dendritic cells. So what is unique about NY-ESO-1? Why do we want to target this antigen? This is because NY-ESO-1 is not found in normal cells, but is expressed by many cancer cells. When we now inject the dendritic cells back into patients, this is what happens. The dendritic cells now recruit an army of killer cells, as well as instruct them to seek out and destroy cancer cells that bear NY-ESO-1. You can see these red dots. Those are NY-ESO-1 on the surface of the cancer cells. So the dendritic cells play a critical role in determining this kind of response. But the story doesn't end there. These killer cells have only a short lifespan. In time, the killer immune cells begin to lose ground. They prematurely call off the attack, as you can see here, and the cancer grows back. So this leads to the second unique aspect of this clinical study. Dr. Pratul Shrikant, a member of our team, made the striking discovery that rapamycin, a drug that's been around for years and used to prevent organ rejection in transplant patients, when used at low doses, converts short-lived immune killer cells into long-lived memory cells. So that with the addition of rapamycin, as shown here, you can see that these cells now come back, they have memory, they're able to remember that cancer cells are bad, they need to be destroyed, they need to be killed. So that instead of expending all of their energy and ammunition upfront, they're able to be strengthened by rapamycin, and they hold on to their potency essentially forever. This hypothesis that we show here has been validated here at Roswell Park in preclinical studies, and we're now going to test it in the clinical setting for the first time. Because Dr. Shrikant's findings represented a remarkable discovery, in addition to patients getting the dendritic cell vaccine that I described, they will also get low doses of rapamycin in order to generate memory and thereby prevent recurrence of disease. Patients with many cancer types will be eligible for this trial, but the tumor has to express NY-ESO-1. Many types of cancer types, such as ovarian, breast, bladder, kidney, melanoma, brain, lungs, gastrointestinal cancers, and so on, will all be potentially eligible providing they meet the requirement of NY-ESO-1 expression. I also want to point out that we, in fact, have conducted a number of phase 1 and phase 2 clinical trials of earlier forms of the NY-ESO-1 vaccine. And the results have been encouraging in ovarian cancer patients. Importantly, we have not seen any significant side effects from those vaccine trials. But the current study that we're talking about takes us to the next level in terms of the long-term effectiveness of the vaccine and with possible application for other disease types. So the third unique aspect of this clinical study that we are presenting today is our manufacturing process. In order to manufacture the vaccine, we developed a highly specialized barrier isolator system, as shown here, that is the first research facility in the US to use this custom-made barrier isolator for vaccine cell production, and in fact, the first in the world to use this system in an approved government-regulated study. At this point, I would like to introduce Dr. Christopher Choi, who was recruited in 2010 from UCLA to head up our new therapeutic cell production facility. Dr. Choi's extensive training in immunology and regulatory science, as well as his 10 years of background experience in cell production, made him the perfect candidate for this job. Dr. Choi will discuss what makes this facility unique and why it is important. Dr. Choi?
I'd like to thank Dr. Adensi for that great introduction. I'd like to thank Drs. Trump and Johnson as well for flying me out to Buffalo from California to take part in this landmark clinical trial. And as Dr. Adensi mentioned, there's many components involved or necessary to complete a clinical trial of this magnitude, of this type. And one of these components is having a special facility or laboratory called a Good Manufacturing Practice Facility, or GMP, where you can manufacture a therapeutic product under FDA regulations and New York State regulations as well. And under the leaderships of Dr. Trump and Dr. Johnson, and in an effort spearheaded by Dr. Adensi and Dr. Hone here, we actually have, President Emeritus, we actually have this GMP facility on site here at Roswell Park. Now, the GMP facility here at Roswell Park is unique because we actually went to the FDA and sought their guidance and advice in the operation and the construction and the design of our facility. We wanted to do it right. And our GMP facility has all the features required by the FDA to produce these life-saving therapies. Not only do we have a state-of-the-art GMP facility, which is monitored for particles, humidity, temperature, pressure 24/7. We also go beyond state-of-the-art. We have this custom-made barrier isolator, which is called the Ex Vivo from Biospirix. Now, this Ex Vivo maintains a class 100, meaning there's only 100 particles of dust per cubic foot within that processing chamber area. And this is required by the FDA. But we go beyond. And as you can see here, the size of the Ex Vivo is that of a skinny minivan. And it has all the components and equipment necessary to manufacture a therapeutic cell product all in one solution. The barrier isolator takes up a smaller footprint than a standard GMP facility, and we believe that the cost of operating and installing this is significantly less than a standard GMP facility. Now, what separates our barrier isolators from others that are out there is the fact that, one, we can precisely maintain control of the manufacturing temperature, and two, we can maintain a precisely controlled atmospheric environment. And we do this by inputting nitrogen, oxygen, and carbon dioxide at any levels we choose to best fit the therapeutic product that we're manufacturing. Now, when we manufacture these potentially life-saving therapies, this DC vaccine, we go through a process which is ex vivo. We take blood from a patient, and once we isolate this blood component from the patient, we take it outside of the patient and manipulate this blood, these cells, ex vivo, outside of the body. After the manipulation, we train these cells, or we prepare these cells to fight cancer. Then after we manipulate these cells, over several days, we infuse these cells back into the patient where they recruit the necessary cells to actually fight the cancer. And as you can understand, or you could tell, the whole process of actually taking out the cells from the patient is probably not a good one. The cells prefer to be inside the body. So we figure, or we believe, that if you mimic conditions that mimic physiologic conditions, this will be better for the cells that you're producing for this DC vaccine trial. And with this, we believe that we not only improve the quality, but we're making an optimal product by also increasing survival. Now, in addition to this, the ex vivo barrier isolator, we expect to be able to maintain a higher level of contamination control than with the standard method of producing a GMP product. And I just want to show here that with the ex vivo barrier isolator, oxygen, carbon dioxide, relative humidity, chamber pressure is all controlled real time. And this is a picture of us working in the ex vivo. Now, as you can see, compared to a standard GMP manufacturing situation, the biggest component or the biggest source of contamination to the product is the person actually making the product. So with the ex vivo, that component is taken away from the equation. So we believe we're making a safer product, not only higher quality, optimal for fighting cancer, but a safer product. We think all is very important. So in conclusion, the GMP facility here at Roswell Park, being the first in upstate New York, allows us to fill a much-needed void. Our facility is available for our colleagues at places at University of Buffalo, Buffalo General, and having this unique cell production facility here allows us to make cell therapy drugs in-house, allowing us to get life-saving medicines to our patients faster. And it all starts with this new vaccine trial. Well, I'd like to thank everybody for being here, and if you have any questions, I'll be taking them now.
Questions for Dr. Odunsi or Dr. Choi?
Do you anticipate that you'll be manufacturing other therapies as well then in the isolator other than this vaccine?
Absolutely, yes.
One of the challenges that has already been posed to the leadership of the Institute is the need to do more than one trial at one time. This will require probably more ex vivo instruments in order to do that. So our development plan over the next few years is to expand this capability so that not only can we do more trials, but also the partnerships with others in the region would allow other things to be done as well of this nature.
And I would like to mention that I've actually already, we haven't even advertised the GMP facility, but word has already gotten out, and I have been approached by a couple of people, not from Roswell obviously, but outside of Roswell, to use the facility for grants and other things.
How will you determine who gets tested?
This will be determined by the treating physician. There are eligibility criteria, and we will make available the protocol. We will make it broadly available through normal channels, through the RPCI website, through the NCI clinicaltrials.gov, so that potentially eligible patients can be identified by their oncologists and sent for the testing as appropriate.
May I follow up? Are these people who are in a certain stage of cancer at that point when you test them?
Yes, depending on their stage as well. So there are two groups of patients. Patients who are at high risk of relapse, who have completed their standard therapies. Potentially, some of those patients could be eligible. And then there are patients who may have what we call minimal residual disease, and some of those could also be eligible. But the details of the clinical protocol and eligibility criteria will be widely available through those channels.
David?
Dr. Choi, you mentioned that we anticipate that the operating costs for this unit will be less expensive than for a conventional clean room where people walk in and the whole room has to be kept sterile. What about the cost of building this or acquiring this? Is it more or less expensive to go with an isolator than a conventional clean room? Any thoughts on that?
Absolutely. In general, a clean room, you don't see a lot of them around in academic institutes because of the cost to building one. I know other institutes have spent tens of millions of dollars. But with this new technology, you basically have a GMP facility in a box. So the cost of making or performing these type of cellular therapy is minimized drastically by, I would say, at least half.
Is there an opportunity in the future then, in addition to treating cancer patients to prevent relapse, that this vaccine could be used to help people avoid getting cancers in the first place?
That is possible. However, so for certain types of cancers, that paradigm already exists. For example, for cervical cancer caused by human papillomavirus, there's a vaccine to prevent cancer of the cervix. For other types of cancers that are not caused by viruses, we are still ways off from developing vaccines to prevent cancer in the first place. However, that is something that potentially is possible in the future.
It's probably also worth pointing out that there exists a commercially available vaccine of similar description that has already been FDA approved to treat men with advanced prostate cancer. This vaccine clearly improves survival but there's a long way to go to optimizing that kind of approach. But I think the proof of principle has been established in human beings, and the trial of Dr. Odunsi and his colleagues will be several steps forward in advancing both treatment of established disease, high-risk patients, and conceivably in the future a preventive strategy.
What is the approximate time frame for the project, the initial trial? When do you expect to see the initial results of the trial?
I think within the next, it depends on when we complete the trial, but once we complete the trial, usually within a year, year and a half of completing the trial, we will be able to demonstrate, hopefully, the hypothesis that we proposed that will be able to generate cells with memory attributes. And we will have some insight also into how the patients are doing. This is a phase 1 trial. It is not an efficacy study. So our objectives are primarily to show that this is safe, it doesn't cause any harm, and that we're able to generate the memory cells, and we will simply observe whether or not we're able to prolong time to relapse in patients.
How many people are going to be a part of this phase 1 trial?
The initial trial will enroll between 18 and 20 patients. Again, because it's a phase 1 study, and once we demonstrate good results, we anticipate that we'll be able to move on to phase 2 efficacy study.
Yes, ma'am.
Across cancer types, those 18 or 20, or is it specific for cancer types?
For all cancer types, the first step, as I said, is the proof of principle, safety, toxicity, generation of memory cells. So once we show that in all the different cancer types, then we'll be ready to move on to a phase 2 efficacy study.
You mentioned earlier that you had already done a couple of trials. I thought you described them as phase 1 and phase 2. What can you tell us about those?
Right. So the previous trials were using less advanced vaccine strategies based on the level of understanding that we had at the time, as well as based on the resources that we have available. But those previous trials, again, in some of those trials, we were targeting the NY-ESO1 antigen. We were simply taking portions of the NY-ESO1 protein and injecting it into patients. We did not go through the elaborate technique of taking out the dendritic cells, growing them to large numbers, and putting back into patients. This is the first time that we're going to be doing that. Nevertheless, even those initial trials showed encouraging results in ovarian cancer patients.
Well, if there are no other questions, I'd like to thank you all for coming and sharing our excitement. Thank you all very much.